Hi everyone, thanks for coming today, it's really nice. Thanks lizard nerds, I saw loads of you in the audience, thank you for coming and representing. My name's James Stroud, I'm at Georgia Tech, I've just started my own lab up there over this past year. I'm recruiting at all levels, so this talk is going to be a little bit about research and a little bit of a pitch as well. And really what I'm going to talk to you about is kind of the research or the topic that my research is grounded in my lab, and that's how we can use field studies to directly measure or attempt to directly measure natural selection in the wild, and how that might explain species diversity at bigger temporal and spatial scales. And so I'm not going to belabor this introduction too much, but this is a really diverse session, and so I wanted to give you a bit of a background into what I did and why I did it, why I wanted to pursue this research. And that's really because a classic way that biologists have thought about the ecological and evolutionary processes of community assembly, of community formation, is by studying adaptive radiations, like this spectacular group of Vangus birds from Madagascar. I always like doing a bait and switch, starting with birds to get the people in the room and then switching to lizards. One way that people have really thought about this community assembly, the way these communities form, has been through this lens, through the kind of evolutionary mechanistic lens of an adaptive landscape. And here what I'm really talking about is how variation in fitness among individuals corresponds with phenotypic variation among those individuals too. It's most typically studied, or it's represented in this kind of multi-dimensional trait space. And I'm particularly interested in how we can apply this idea, apply this concept to entire ecological communities. So for example, the trait space in these Vangus birds may represent variation in phenotypic traits, such as beak shape or beak size or wing length, traits that might correspond to occupying different ecological niches. And we'd visualize the distribution of fitness on this adaptive landscape as this rugged multi-peak surface, right? Fitness here representing biological fitness, so survival probability or reproductive success or whatever it is you want to measure would be highest here in the very peaks and lowest in the valleys within the surface. And we'd expect species in the wild to correspond to these independent regions of high fitness, these independent fitness peaks. And the existence of these fitness valleys surrounding these peaks really limits these species from diversifying. And so like the stranding of species on these independent fitness peaks really underlies or drives the observable structure of ecological communities in the wild. And so the existence of the adaptive landscape as a metaphor has really provided a fundamental way in which we think about life on Earth. However, despite adaptive landscapes representing this incredibly powerful conceptual heuristic for the ecology and evolution of species diversity, the concepts largely remain metaphorical at the community level, and this is surprising because the utility of it in evolutionary biology is really important, right? Potentially underpinning kind of everything about what we understand about the evolutionary origins and the processes involved in determining community structure. And so a handful of pioneering studies have really attempted to collect empirical evidence to test this hypothesis, and there's been two main approaches to doing this. First is fitness surfaces can be estimated from trait proxies where populations are sampled at a single time point. So here in this community of sailfin fishes in Sulawesi, Indonesia, about 1,000 fish of these 5 species were caught, and individual fitness was estimated from relative liver weight of each individual. And from those data, it produced this incredibly complex multi-peak or multi-species fitness surface. Alternatively, fitness can be measured directly among individuals at discrete time points. For example, here in this a series of some of my favorite studies, Chris Martin estimated fitness in this 3-species community of Caribbean pupfish. And this was really an exceptional study in that he produced hybrids of these 3 species in the lab and introduced them to experimental pens in the wild. And so he was explicitly testing this multi-peak hypothesis. Producing these hybrids really forced the existence of phenotypes that we might not naturally find in the wild, right? So you have the chance to experimentally probe the landscape in places that you might not be able to measure naturally. Well, I was always curious if it would be possible to just do that, to identify if these same types of patterns could exist just using natural variation. In other words, could we identify fitness differences? Could they be detected among the phenotypic variation that just exists naturally in the wild in kind of complex communities? This is what evolutionary theory would predict, but it's a prediction that's been very rarely tested at the community level. And so I tried it. Using a community of 4 anolis species, each one here that are ecologically and morphologically distinct, right? So in this group, we refer to them as separate ecomorphs. They pretty much occupy kind of distinct microhabitats, separate niches in the environment, And that, in a very robust way, determines community structure among these communities across space. And through a massive mark recapture research effort, I wanted to measure fitness in this wild community and see if we can estimate this type of selection surface, this type of selection landscape. Specifically testing this hypothesis that different species in this community, those that occupy different niches, are also occupying different independent fitness peaks on this landscape. But despite the importance of this, it's really rare to attempt this because it typically represents a huge amount of work. And so for this research project, I caught nearly 2,000 lizards. Here they are all plotted in multivariate morphological trait space. The two axes in these trait space represent morphological traits that we know are adaptively significant in anoles, and so traits that we think might influence survival, important fitness-related traits. And we generate this morphological space by systematically collecting a bunch of trait data, both skeletal using my lab's new fancy portable X-ray machine, which we bring to the field with us. This is in my field lab in Miami just in May this year, as well as a bunch of other stuff like cell phones or scanners to collect non-skeletal trait data. And as I said, these traits are really important in the sense that we have a foundational knowledge of their function. And so we can see if selection's acting on them, we can kind of interpret them through an ecological or fitness lens, an ecological or functional lens. And we can estimate survival using these uniquely coded tags injected underneath the skin in these lizards, only visible under a UV blacklight, and so they have no discernible effect on survival. And as I said, it's a ton of lizards. This is a video I just snuck in earlier this morning. I thought it would be cool to show. From May this year, when we went back to sample this community, this is a result of just maybe 1.5 days of catching right at the start of the project. Anyone that knows that does mark recapture stuff knows you catch 90% of the lizards or animals in the first 10% of field work, and then the rest is the most annoying individuals that take ages. And so I wanted to include these slides because, as I said, I'm recruiting and I want everyone to see kind of the cool stuff that underlies these patterns. This is the nuts and bolts. This is how we collect these data. We also collect tissue samples from every single individual so we can genotype them and we can start to assess pedigrees through time. And so back to the lizards, a lot of blood, sweat, and tears went into producing this really, really simple plot of little grey dots. Each point here represents the position of each individual lizard in this community in this multivariate trait space. And through this exhaustive mark recapture sampling, we can see who in this community lives and who dies, allowing me to estimate how fitness is distributed across this entire community, across this entire trait space. But first, an important part of this is you might notice that there's some structure in the distribution of these traits. And that's because this is a community, right? This is an ecological community. These 4 species, as expected in any community, do form loosely distinct clusters in trait space because they are different. This is what we would expect. Each represents a separate niche specialist, a separate ecomorph class, and so traits in each species are adapted for the divergent portions of the environment that they occupy. And so I first wanted to know, given this interspecific structure in trait space, how likely is it that a fitness surface just estimates peaks in each of these clusters just through a random survival of individuals? And so to do this, I developed a null model approach using permutation tests where we just randomly reshuffle the survival of individuals, and then we can estimate the shape of the fitness surface that each of these random simulations produces using thin plate splines. And so here, the histogram on the left shows the distribution of these random simulations, distribution of the complexity of the fitness surfaces. The vast majority of these fitness surfaces are completely flat, like on the right-hand side. The x-axis here is the effective degrees of freedom. It basically describes the topographical complexity of the fitness surface. And as I said, most are really flat. And so this is where the empirical surface lies. It's a surface that would have been incredibly unlikely to have arisen by chance, by random survival, right? In fact, the curvature of this surface is more complex. It's more rugged than around 99% of all other configurations of individual survival. And so what does it look like? Looks like this. And this, to me, was beautiful, right? This is such a nice result to get. It's complex, which is cool. And it suggests that there's some structure, there's some inequality to the distribution of fitness in the community, but it doesn't really tell us what we want to know yet, right? What we want to know is, does any of this complexity align with what we know about this community? Do species in this community correspond to these independent peaks? And that's really the million-dollar question. And so this is exactly what we see. Very much to my own excitement, each peak was occupied by a different species. The circle pins here reflecting the position of the mean phenotype of each species on this surface. And you can see here on the right, the distribution of fitness as it relates to the boundaries of each species and trait space. And so really here, each ecomorph is sitting proud atop its own fitness peak in this in separate regions of the adaptive landscape. And so not only does each species occupy independent fitness peaks, but there exist these fitness valleys between species. And this has been a really hard thing to test in the wild. And so this really provides, in my opinion, excellent evidence that natural selection is acting to maintain these species as divergent phenotypes through time. Any individual lizard, any individual organism with a phenotype intermediate to two species finds itself located in these valleys of low fitness. And so this pattern would suggest that the fitness of any individual is a function of how similar it is to the mean phenotype of that species, right? Complicated way of saying we'd expect stabilizing selection to be operating within each species. And so we can test that explicitly. Here are two ecologically divergent species in this community, very distinct morphologically and ecologically. On the left here is the green trunk crown anole, and on the right is the brown trunk grounds anole. One's an arboreal specialist, one's a terrestrial specialist. And we can see here on the bottom right that they occupy different peaks on this complex community-wide fitness landscape. In the top right here, we can see the distribution of survivors versus non-survivors of each species independently. And in these main figures here, I've fitted fitness functions using cubic splines to estimate the distribution of fitness as it relates to their similarity to a mean phenotype. And so what we'd expect is that fitness, or here survival probability, would decrease as you become increasingly dissimilar to the species mean. And so that's exactly what we see here. A stabilizing selection acting to maintain this central phenotypic optima seems to be independently acting and independently maintaining these two species as divergent phenotypes, right? Each isolated on these independent fitness peaks as individuals that possess increasingly dissimilar phenotypes experience fitness costs. Of course, this is just one way of testing for stabilizing selection. I did it a bunch of other ways. You can read the paper for those details. But the punchline is really that all the different methods had the same sort of result. There was strong evidence, strong effect for stabilization in this form of selection. And so this is what the pattern looks like for each species independently. These are fitness surfaces just for the species, not from the community level, projected into 3D space. And you can see these surfaces resemble a peak or a hill or a mountain or whatever metaphor you want to use. But really the main part of this to focus on is that fitness declines, right, in the most extreme, the most peripheral phenotypes. But the result of this is the accumulation of multiple sampling sessions through time. And it was such a strong result, I just figured that these independent periods that produce this, that accumulatively produce this, were just doing the same thing, right? It was just lots of smaller variations of this. But when we probe that temporal landscape, we can see that selection actually varies dramatically. It fluctuates wildly through time. In these 5 time sampling periods, we very rarely see periods of stabilizing selection like we did down there in the bottom right in winter 2015 for that brown anole. In fact, what we see is just many periods of linear selection. The fitness peak for each species bounces around through time. Sometimes it isn't even discernible from the trough of the fitness valley. So periods of weak or relaxed selection, at least in these morphological traits. And so the next question in my lab is why? I don't know if any of you went to the sessions on the predictability versus the unpredictability of evolution, but I think it's been a really good part of this conference. And for me, one of the issues with that session has been the fact that we don't really have a very good understanding of the ecology of natural selection in the wild, right? We often identify the pattern, but understanding the process and why some things have higher fitness versus lower fitness is a really, really difficult thing to study. And so that's what me and my lab are trying to do using this same community, understanding what drives these fluctuations through time by quantifying in detail the abiotic and the biotic environment, and also many new measurements of individual phenotype. So allowing us to measure fitness as it relates to not only morphology, but ecological and genotypic and performance-related traits. And this study has been going on continuously, or nearly continuously, thanks COVID, since 2015. And so now we have some really nice longitudinal data sets of selection dynamics within a community framework. And so in my lab, that's really what I'm interested in doing. I'm interested in using field studies to try to transcend scales, so understand the full process from how the fitness of individuals might dictate patterns at the community level and how we can elevate that to understand much broader patterns of biodiversity across space and time. And so with that, I'll say please get in contact with me if any of this interests you. I'll be around all afternoon and at the Super Social this evening, so grab me whenever you like. I don't know if we have time. I'm sorry, I don't want to bump into the second speaker, so just come and grab me.
I think I'm just going to get started. So, hi everyone, thanks for coming to the session. My name is Zhang. I'm a PhD candidate in Dr. Regina Balcom's lab at the University of Michigan. And today I'm going to tell you a little bit about some of the preliminary results from the second chapter of my dissertation on the fitness, costs, and benefits of resistance to glyphosate and herbivory in an agricultural weed. Plants face multiple stressors that are all occurring at the same time. In this illustration by Maria Sibylla-Berrien, we can see some examples of these stressors, including environmental stressors like precipitation or temperature. We have here insect herbivores that might damage and consume plant tissue. And we have plants that are existing in close proximity to each other, so competing for shared resources. But all of these stressors that we see, at least here, are natural stressors. But of course, we know that humans, we have dramatically altered the planet, and we have introduced anthropogenic stressors as well. And this anthropogenic change can also impact plant evolution. One of the main ways that we humans have altered the planet is through industrialized agriculture. And especially in the past century or so, another main way that agriculture has impacted plants in particular is through the widespread use of chemical herbicides. Here I'm showing a map of the use of Roundup, also known as glyphosate, in the United States just in 2019 alone, and you can see it's all over the place. And these chemical herbicides, they act as novel selective agents on these plants, and that's because herbicides are really good generally at what they do, right? They're made to kill plants, and they're really good at it. So they exert really strong selective pressure on these plants to evolve some form of resistance to them. But this evolution of herbicide resistance doesn't just occur in a vacuum. It's happening in the context of other stressors as well, such as insect herbivory. And plants and insects have co-evolved for hundreds of millions of years to the point where plants now have all sorts of defenses against insects, including chemical defenses, physical defenses, as well as mutualisms with other organisms. And few studies have really looked at how herbicide and herbivory impact plants at the same time. One of the few studies that have done this is by Gassman and Fatuma back in 2005, and they were looking at Amaranthus hybridus that was susceptible and resistant to the herbicide triazine. And they were doing this in the presence and absence of the specialist herbivore, the striped leaf beetle. And we can see from these plots that the triazine-susceptible plants, which are represented in the open circles, or excuse me, the triazine-resistant plants represented in the open circles, they have lower floral and vegetative biomass in the presence of these herbivores. So we see here a pretty clear example of a trade-off between resistance to herbicide and resistance to herbivory. And interestingly, this is a case where that trade-off, that cost to herbicide resistance only really shows up in the presence of that secondary stressor in the form of herbivory. So, why are we thinking about costs at all, right? So to take a step back, fitness, costs, and benefits really help shape plant evolution. As evolutionary biologists in general, we're really interested in how and why trait variation persists in with both within and between populations. And one of the possible explanations for this is the limitations of resource allocation, right? The idea is that plants have a limited pool of resources that they have to put towards multiple processes, in this case, resistance and fitness, such that if they put a lot of energy towards resistance, they're not going to have as much to put towards fitness. And folks have looked at costs and benefits of plants, of resistance to a variety of different stressors, including different types of insects, different types of herbicides, fungal and microbial pathogens. And generally, they've done this by assessing benefits in the presence of the stressor and costs in the absence of the stressor. So the idea is, in the presence of the stressor, a plant that's more resistant to it would be more fit than all the other ones that are killed and more susceptible. Whereas in the absence of that stressor, this resistant plant then accumulates this fitness cost because it just didn't put as much energy towards that. But in general, these studies have focused on a single stressor at a time, right? But as we talked about earlier, plants don't exist in a vacuum. So what I'm broadly interested in is how do plants adapt to these multiple stressors that are all occurring at the same time? And more specifically in this project, I'm asking if there are fitness costs and benefits that are associated with herbicide and herbivory resistance. And in particular, the system I am using is the Roundup and Morning Glory system. So the common morning glory, Ipomea purpurea, it's an agricultural weed. Past work has shown that it's evolved resistance against insect herbivory, and also that it has natural varying levels of resistance to glyphosate. And some of the genes associated with glyphosate resistance in this species include cytochrome P450s and glycotransferases that have to do with detoxification pathways, as well as phosphate transporter genes that have to do with translocation pathways. And so for this experiment, we did a common garden study using 1440 plants from 45 family lines with replicates across multiple environments, which I'll talk about in just a little bit. We applied glyphosate to half of these plants, and we scored the damage as the number of damaged leaves over the total number of leaves, so the proportion of leaves that were damaged. We assessed herbivory damage as the percentage of tissue missing from 4 leaves per plant. And we are calculating resistance as 1 minus damage, so the inverse of damage. And our fitness measures in this study is seed number and seed weight, so the quantity and quality of these seeds. In terms of the actual experimental setup, the colors are a little faint here, but we basically did a factorial setup where we have different environments with different stressors present. So 2 of these environments have glyphosate present, and then 2 of the environments have herbivory present. And we got rid of herbivory using the insecticide spinosad on those bottom environments right there. And this setup lets us answer different questions in each of these different environments. For example, in that top right-hand corner, where both glyphosate and herbivory is present, we can assess if there are fitness benefits to resistance to either stressor. In the environment where only herbivory is present, we can see if there is a cost to glyphosate resistance, and if there's a benefit to herbivory resistance. In this third environment where only glyphosate is present, we can ask if there is a benefit to glyphosate resistance and a cost to herbivory resistance. And lastly, in this last environment where neither stressor is present, we can see if there's a cost to either type of resistance. And so if we're just thinking about glyphosate alone, we would conclude that there is a fitness benefit to glyphosate resistance if we see a positive relationship between resistance and fitness, whereas in the second environment where glyphosate is not present, we can assume that there is a fitness cost if we see a negative relationship between resistance and fitness. So moving on to the results, I will be showing you some selection analyses for both types of resistance. This plot on the left-hand side will be showing you the selection analyses for glyphosate resistance, whereas on the right-hand side, I'll be showing you the results for herbivory resistance. In this bottom right-hand corner, I'll be showing you which environment we're in. In the top right-hand corner, I will show the question that we're asking, so whether we're assessing for the benefit or cost of either type of resistance. And then lastly, relative fitness will be shown in both seed number and seed weight. So moving into our first environment where we have both glyphosate and herbivory present, in terms of seed number, we can see that there is a benefit to glyphosate resistance. We see positive selection for that trait, but we don't see it for herbivory resistance. Each of these points are family line averages, so this is genotypic selection that I'm showing here. And if we incorporate seed weight, now these points are color-coded by their average seed weight per family. So if we do a regression line with seed weight on glyphosate or on either type of resistance, we can see that there is now positive selection for both glyphosate and herbivory resistance in terms of seed weight. And in this particular environment, seed number and seed weight are correlated. In the second environment, where we only have herbivory present, we see that there is no cost to glyphosate resistance in terms of seed number. But surprisingly, we see negative selection for herbivory resistance, during which we might expect to see a benefit of it because that stressor is present. So that was really interesting. If we look at it in terms of seed weight, we don't see any cost or benefit to either type of resistance. And in this environment, seed number and seed weight are not correlated. In our third environment, where only glyphosate is present, we don't see any signs of selection in terms of seed number for either type of stressor. But if we look at things in terms of seed weight, we see potential stabilizing selection for glyphosate resistance, but no cost or herbivory resistance. And in this environment, seed number and seed weight are correlated. And in our last environment, where neither stressor is present, we don't see any costs across the board in terms of either seed number or in terms of seed weight. So those were a lot of plots that I just showed you, so just to sum everything sort of up in this environment. In this first environment where both stressors are present, we see fitness benefits to both glyphosate and herbivory resistance in terms of seed weight, and we certainly see benefits of glyphosate resistance in terms of seed number. In this second environment, where only herbivory is present, we don't detect any costs for glyphosate resistance, but very interestingly, we do for herbivory resistance, which again, we didn't expect, and I'll talk about that in just a moment. In this third environment, where only glyphosate is present, we see potential stabilizing selection for glyphosate resistance in terms of seed weight, but not in seed number, and there were no costs detected for herbivory resistance. And in our last environment, where neither stressor is present, we don't see any cost for either type of resistance here. So really just to zoom in on that herbivory resistance bit, right? So in the presence of herbivory in both of these environments, if we have also glyphosate present, we see positive selection for herbivory resistance in terms of seed weight. But once we remove glyphosate, we see negative selection for herbivory resistance in terms of seed number. So what this suggests is that glyphosate is somehow mediating these costs and benefits to herbivory resistance. And we're still digging into how this is happening and why this is the case. These are fairly preliminary results. But interestingly, this is in contrast to that Gassman-Fatuma study I showed you earlier, which is one of the few other studies that's really looked at herbicide and herbivory resistance concurrently. In that study, they showed that it was herbivory that was affecting herbicide resistance, whereas we kind of showed the opposite. So that was kind of cool. So in addition to digging more into this particular relationship of how glyphosate is mediating herbivory resistance, next steps also include conducting germination assays of these field-collected plants to see if we can detect any fitness costs to glyphosate resistance. Past work in the lab done by Van Enten et al. Has shown that if populations of morning glories that have higher levels of glyphosate resistance do show lower rates of germination. So I want to see if that is borne out by the seeds that I've collected from the field from plants that were actually affected by glyphosate. And with that, I want to thank everyone in the lab, everyone that's helped in the field, and my funding sources. I'm happy to take questions. Thank you.
Okay, so hi everyone. My name is Jorgen Soraker, or this, that my conference badge chose to call me for the first couple of days. I'm happy to take any challenge to actually pronounce that after my talk. I am a PhD student at Edward Gray Institute at the University of Oxford, and I focus on natural selection. And today I'm going to talk to you about a project that I'm working on, which is about the spatial synchrony of selection. So first, just over to the problem, natural selection, which we can define as any covariance between phenotype and fitness. Well, we have thousands of estimates of natural selection today, and they vary for a lot of different species, for a lot of different traits, and such a major undertaking in ecology and evolution have been to understand how selection varies, both in time and in space. And it's been done quite a lot on this, and big meta-analysis and so on, and that have shown that we have a lot of variation, both spatially and temporally, in selection. But it still remains rather unclear how selection for a specific trait among different populations co-vary. So how does it co-vary both in space and in time? So to illustrate this for you, here I have 4 great tit populations. Spoiler alert, I work with great tits and blue tits, so you're going to hear a lot about that. Imagine that we have these 4 populations, and I put up a ruler, and the red line is supposed to reflect the selection estimate. Well, is it so that all these populations experience high selection one year, for example, and that this can lower in all of the populations the next year? So basically, like the tide rises and lowers. Or is it more like a wave function, that populations closer to each other tend to experience more similar selection regimes? So let's say that it can vary like this. Okay, this leaves us with 2 specific questions. First, how does selection for a trait co-vary among different populations of the same species for the same trait? And second, are we actually able to identify some of the drivers of these patterns? So if it turns out to be like the wave thing, which, spoiler alert, it turns out to be, are we actually able to identify what are causing these waves? Can we actually identify these ones? So these are the problems that I want to work with. And one way we can actually try to do this is to use something called spatial synchrony. So I asked ChatGPT, how would you describe spatial synchrony to a 5-year-old? No offense. And I had to translate over from Norwegian again, but it basically said that closer things are more similar than things further apart. And I think that's actually quite a nice summarization of it. More academically, it's spatial scaling of correlated traits. And this method was actually developed for population dynamics to see how population numbers increased and decreased with each other and among space. And the really beauty of this method is that you can include any explanatory variable that you want, and you can see how it actually changes the synchrony. So you will get a pattern like you see down here at the right. So for each population, we make every pairwise combination of different populations, and these have repeated measures, for example, of a trait, for example, population size, or in my case, selection, and we estimate a correlation in selection among these populations. And we are mainly interested in 3 parameters in these models. So you have the correlation at distance 0. So imagine 2 populations that are right next to each other. How correlated are these ones in the trait of interest? And then you have correlation at distance that goes towards infinity, which is sort of the baseline fundamental correlation that exists out there. And then you have a parameter that basically determines the shape of this function. So it's a smoothing or a distance parameter. These ones tell you actually at what distance do the trait of interest remain correlated among populations. So we hope that we can actually use this method not to look at population size and so on, but actually to look at selection. Because we have a lot of population, especially in Europe, where we estimate selection for each year. And then we hope that we can actually use this method to investigate the spatial synchrony of selection. So I contacted a lot of different data holders, a lot of different populations that worked on gray tits and blue tits. And I asked if they wanted to participate, and almost 40 populations said yes, which is just incredible. It's one of the major advantages of working with species like gray tits and blue tits. So many actually study them. So I asked for populations that had reproductive traits, like onset of breeding or lay date, and the clutch size, with the corresponding number of fledglings that were produced for that specific clutch. I wish I could show you the results of all these 40 populations. However, there is still a delay in handing over data and so on. So today I will show you some results with a subset of these. This is only for gray tits, and it's from 2000 and onwards for 19 different populations. And this is a really beautiful base R map that you can see where these populations are spread out. So what these models does is that it basically gives you some general outputs. So we find, for example, if you consider all the populations across all the years, we find selection for larger clutches, but this is nonlinear, so we have support for a squared effect. We do not find overall selection for early laying, which is quite interesting because it's widely hypothesized to happen. And then we also included a correlation between lay date and clutch size to allow for correlated selection, which basically shows that there is correlated selection for larger clutches when you lay early, so at the combination of traits. So this is not as bad as it looks. This is just to show you the LME4 model. What I just want to highlight here is that you allow each trait of interest to vary within the random effects. So you basically have a random slope model. That's it. It's not as bad as it looks. And if you do the decomposition of these random effects, you can then combine and get the selection estimate for each population for each year. So that's what I did. So here, each point here is selection estimate for one population for 1 year, and you have the population over there. If you're able to crack all the codes, I will be really impressed. But if you look at, you have selection for lay date or breeding onset on the left and for clutch size on the right. And you see, I told you that we don't have overall selection for earlier laying, and you see it varies quite a lot around 0, but for clutch sizes tend to be quite positive all the time. There is also another important thing to notice with these graphs, and that is that for lay date, they seem to not only co-vary, but they are really clustered together. But for clutch size, the lines seem to be more separated. So each line is a population. So in some populations, you basically have more consistent higher selection or stronger selection for clutch size, for example. And also, when you use this model, you're also able to decompose the variation that you find in selection estimates. And this shows that for clutch size, we have a lot of the variation is explained by the different populations. Also, just want to mention that you can do the exact same thing with correlated selection. I don't want to dive too deep into it, but basically it varies around 0, which is quite interesting because this basically determines the shape of the different surfaces and so on. Don't want to spend too much time on that by now. Remember, this is all rather preliminary results. So, then what about the synchrony? I said I wanted to estimate this synchrony stuff and so on. I wish I could show you some really nice graphs with really nice lines and beautiful uncertainty intervals and so on. However, I made these plots yesterday, so bear with me of the ugly base R plots. So this is what it looks like when here each point is a pair of populations. So it's the correlation among these specific pair of populations. And this is for selection for lay date. And you see that it basically gives you, or will give you the same shape that we saw on different plots earlier, where we have a correlation at distance 0, which is around 0.36. And when you go towards infinity, it's like 0.06. And this is quite interesting because if you do it for lay date alone, you will find a correlation at distance 0, which is basically up around 0.6, 0.8. So you have a much lower synchrony in the selection of the trait than for the trait itself. And when you use this instead on clutch size, you find an even lower synchrony. I told you a few minutes ago that it seemed to be more variation in the selection estimates that are due to consistent differences among the populations. So that might be some of that result that we actually see here then. So that basically answers part of the first question, like do we have spatial synchrony in the selection estimates? And we do. And then this is what we want to do next, and that is to dive into the drivers of selection for these two specific traits. And we have really high interest in population size and in some environmental drivers. For example, we hypothesize that high temperatures can lead to stronger selection for earlier laying, for example. So there are different ways you can do this to identify how these potential drivers affect the synchrony. But some colleagues in Trondheim are now working on a new method that is actually, it looks a bit awful, but it's actually quite intuitive. Let's say that you have an environmental variable, for example, temperature, and you do the spatial synchrony in this. So places that are closer to each other tend to experience more of the similar temperature than populations further apart. That makes sense. We know that that exists. Then you can just estimate the covariance between the selection estimates and this environmental variable. And if the different populations co-vary over time, then this can itself be a part in driving the synchrony of selection. So if you have synchrony in the environmental variables that is then affecting the selection estimates, then that can by definition be a driver of the different selection synchrony. So just some take-home messages. There is a lot of focus on natural selection and estimating it, but it's still quite a lot that we don't know, especially about the spatial temporal variation in selection. We think that spatial synchrony can be a nice platform to actually address such questions. And I think we've shown so far as well that the synchrony you obtain depends a lot on what traits you're looking at. So you need to pay attention to that one as well. And we hope at least that we can disentangle some of the drivers of synchrony in selection. At this conference, I've seen a lot of people with really nice and fancy acknowledgement sites. None mentioned, none forgotten. I just want to say thank you. Thanks a lot. And I'm happy to try to answer any question. Yes?
I wondered what kind of environmental data you have, since these are all different locations and you're getting them from different people. Obviously, there's some available online, but I wonder if there's any environmental driver that you think might be responsible, and if you have the data for that, if it's something unique maybe.
Yes, this is a good question. So it really depends on the trait you're looking at. So we, for example, hypothesize that temperature will be more deterministic for selection on lay date than for clutch size. And we have really nice databases for this across Europe. And a previous student at my lab recently dived into, are these databases reliable? And they found some other methods that we can actually extract it and so on. So I really just build upon what they did. But we do have available information both from the data holders, or some of the data holders, and more like large-scale environmental data.
I know some populations have little variation.
Yep.
So I wonder if it would be not just the temperature of whatever date those were taken, but temperature variation.
Exactly. That's a really good comment. And what we realized quite early when we started with this is that how you do the different standardizations really ask different ecological questions. Because what I've done now is just basically the raw values of selection. But maybe it's more interesting to look at the relative change in selection to the relative change in temperature. I completely agree, and definitely it's a good point. And at the moment, we hope to look into both. What we'll end up doing, we'll see. Yes?
Why are the correlations bounded at 0? Maybe I missed this at the beginning. Why don't they go negative?
Oh, they can go negative.
You never found any, though?
So this is just something from another study, and you do find negative values as well. But in the spatial autocorrelation model that you use, if you use the non-parametric model, then you have to just assume that they're positive. So in some cases, it made biological sense to have negative correlation. But in some of them, especially for some of these questions, it may not be that relative if they're actually negatively correlated, especially when you look at the explanatory variables. So it depends. We do allow them to have negative values, but it really depends if you use a parametric or non-parametric spatial autocorrelation function afterwards. So in most of them, we just bound them to be 0. And here I just cut the figure because it was too big. Yeah. Yes?
Do you think the blue ticks are going to show the same pattern?
They tend to show like weaker selection for specifically, especially for clutch size in some of these populations. So I think they can vary. I think there can be differences between the two species. And also, like, this is a subset of the data. I'm not that worried about the subset of the different populations, but I'm more worried about the subset of the time period. So it's from 2000 onwards, and we know that, especially in the 90s, in a lot of these populations, it was strong selection for early lay date, for example. So it might be that the overall model output can be more sensitive to the temporal subsection. But overall, these are quite similar species with a lot of similar environmental drivers. But that is also why we include both of them, because it will be really interesting to see, do they actually vary? Okay, then I think my time is up. Thank you.
Hello, everybody. I'll let folks settle in a little bit, but my name is Alex Mauro, and today I'll be talking about how a trade-off constrains the realized niche of the Trinidadian guppy. And I first want to put up my acknowledgements because this was a large team effort, and I really want to shout out Julian Torres-Dowdell, Craig Marshall, and Kendall Zeller. So understanding what factors determine species range limits has long fascinated evolutionary biologists and ecologists alike. And the question of what sets present range limits can often be answered with a thorough understanding of the ecophysiology of an organism. But what prevents range expansion, in the absence of dispersal limitations, becomes a question of why can a species not evolve to persist in the conditions beyond its current range? Or in other words, what is constraining the realized niche of that species? And this question has been deemed the evolutionary problem of range limits. And to demonstrate what I mean a little bit more thoroughly, let's return to this concept of the ecological niche. So the fundamental niche is the full range of environmental conditions an organism can tolerate. This black species of fish can tolerate some combination of the abiotic stressors of salinity, temperature, and pH. And this can indicate where we might find it distributed in nature. The realized niche, though, is where we actually find it. It's a subset of that. It's the actual range of environmental conditions we find in the organism, and it's tolerating in nature. An ecological niche theory predicts that a mechanism by which the fundamental niche is constrained down to the realized niche is through antagonistic biotic interactions. So here we can see that that dark blue combination of abiotic stressors combined with this negative interaction with the green species of fish is constraining the realized niche of the black one and setting its range limit. And indeed, that combination of abiotic stressors and negative biotic interactions pose a challenge, and that ecophysiological challenge can explain a lot of current range limits. But it does not necessarily pose an evolutionary challenge. unless the traits underlying abiotic tolerance and performance during biotic interactions trade off. And then they can serve as an evolutionary constraint to range expansion if this trade-off leads to fitness in the home environment, coming at the cost of fitness in the away environment, and there's a negative genetic correlation between the traits. So there's a genetic basis to this trade-off. And evolutionarily speaking, this means that adaptation to the current home environment would come at the cost of your ability to succeed in that away environment. And my colleagues and I went out to test whether this trade-off hypothesis could serve as a solution to the evolutionary problem of range limits in the Trinidadian guppy. Specifically asking the question, does a trade-off between salinity tolerance and competitive ability constrain the realized niche? To go through this investigation, there'll be 4 parts. First, we'll look at the distribution of the guppy as it relates to niches, the effect of tolerating salinity and competition on fitness, evidence for a genetic basis to our trade-off, and physiological evidence for why this trade-off exists in the first place. So all across Trinidad, these 2 species of Facilidated are distributed in a very, very distinct manner. So our focal species, the Trinidadian guppy, and a potential competitor, the swamp guppy. And to demonstrate what I mean, let's zoom in on one river. So there we find that the Trinidadian guppy is always restricted to fresh water, whereas the swamp guppy is mostly found in brackish water, but the 2 overlap in a small region of fresh water near that salinity flux point. And the consistency of this pattern is striking, but what is even more striking is that we know the Trinidadian guppy can survive in brackish water and in seawater in the lab and in other parts of its native and introduced range. And so it begs the question, why specifically in Trinidad are they restricted to fresh water? Or in other words, why is the guppy's realized niche constrained to fresh water here in Trinidad but not in other parts of its range? Now, just based on the distribution of these species along the salinity gradient, we hypothesized that it was a combination of brackish water and the presence of swamp guppies that was affecting Trinidadian guppy fitness in nature. And to test this, we did a transplant experiment. We had cages across a river in Trinidad, in freshwater and brackish water, and within them, Trinidadian guppies were either with conspecifics or they were with swamp guppies. And we looked at survival rate over 3 weeks. And this is what we found. So we have survival on the y-axis, our 2 salinities on the x, and then who the Trinidadian guppies were competing with in the dashed or dark lines. So first off, when Trinidadian guppies were with other Trinidadian guppies, salinity didn't play a big major role, shown by that dark black line. These fish truly are euryhaline. Salinity did play a major role, though, when they were with swamp guppies. There you can see that survival is greatly reduced in brackish water and in the presence of swamp guppies. But what is it about this combination of this other fish and salinity as such a big factor in survival? Or in other words, what was going on inside these cages in nature? To answer this question, we essentially brought those cages in from Trinidad into the lab so we could observe the behavior and this interaction between these 2 fish as it related to salinity. So we looked at F2 adult male fish, and we measured aggression 7 times over 3 weeks, as these 2 different individuals and species competed over a localized food source. And I'll just show you now our interspecific results. So now you can see the difference in aggressive behaviors between a Trinidadian guppy and a swamp guppy, and it's oriented in such a manner that positive values indicate the Trinidadian guppy is dominant, and negative values would indicate that the Trinidadian guppy is subordinate. So in freshwater, you can see that they were more or less equal competitors. But in brackish water, again, a different story. The swamp guppies were dominant. They were kicking the butt of the Trinidadian guppy. So these results apprise with a behavioral mechanism that these fish were actually competing. And we can better understand why this combination of brackish water and swamp guppies was so detrimental to the Trinidadian guppy. But it's still unclear if salinity tolerance and competitive ability in freshwater actually trade off. And so to investigate that, again, our colleagues and I brought the fish back from Trinidad into the lab, and we conducted a split sibling breeding design. We measured juvenile growth rate as a fitness proxy on F2 Trinidadian guppies, as they either developed in freshwater with swamp guppies, representative of their home environment, or in brackish water alone, which would be indicative of a potential away environment. And here is what we found. So we have juvenile growth rate now on the Y, and our 2 different treatments on the X, our home treatment and our away treatment. Those dots are siblings, so those lines are family lines. And what we found was that families that grew worse in one environment tended to grow best in the other, indicative of a trade-off. Furthermore, based on the rank change in the family orders, we found support for a genetic basis to this trade-off. In a broader evolutionary context, this means that involving improved tolerance to salinity would come at the cost of your ability to compete in freshwater. But taking a bit of a step back now, why might these traits be genetically correlated in the first place? And could knowing more about the physiology of this trade-off and these traits help us understand it better? So in the final portion, what we did was try to first synthesize what we knew about these two different traits, osmoregulation and aggression in fish. And there's a wealth of wonderful literature on this. And I'll kind of summarize what we found here just briefly. So when teleos fish are osmoregulating in brackish water, it's a complex process. You can see that it involves organs all across the body. But critically, it is dependent on the excretion of excess ions from the gills, and that is mediated by, a hormonal cascade beginning in the brain. When we look at the brain in terms of aggression and see what's going on, we see a lot of the same players. We see AVT, somatostatin, cortisol, growth hormone. And when you kind of overlay these two networks together, why a trade-off might exist becomes a bit more clear. For instance, if we were to highlight this pink pathway, this hormonal cascade kind of starting with somatostatin, you can see that it downregulates growth hormone. Growth hormone normally increases aggression, so that would decline aggression. Somatostatin increases cortisol, which is critical for excreting ions and salinity tolerance, but cortisol has a direct effect on decreasing aggression in fish. So we then wanted to test to see if this hypothesized network was actually existing in our species. And to do that, we conducted a gene expression network study, and I'll kind of walk you through the steps. So first off, we just replicated what we had done before. It was the same behavior setup, except this time, after 24 hours in our conditions, we sampled the trinating guppy brains after we had also measured their aggression at once. Then we sequenced it using a tag-seq protocol. And then we did a multi-step network-based approach, where the first step was trying to subset our genes to just those important to salinity tolerance. So we looked at all the genes that were differentially expressed across salinities while controlling for competition type. We then used WGCNA, or weighted gene co-expression network analysis, to put these different genes into co-expressed gene modules. And that offered a big advantage because we could then correlate the eigenvalues of these modules with our trait of interest, aggression. And because we had selected our input genes based purely on their role in salinity, a significant correlation with aggression would provide evidence that those two traits share a similar group of genes or a similar group of co-expressed gene networks. And indeed, when we do that, we see that our largest module has a negative correlation with aggression. Moreover, we see a lot of the same players and pathways that we had hypothesized a priori. So again, that kind of somatostatin cascade was supported. And overall, this network analysis provides evidence that hormonal pleiotropy might be underlying this trade-off that we're observing in nature. So sort of to wrap up what we learned from 4 different studies done by a whole team of researchers, understanding the distribution and abundance of species is a goal in ecology and evolution. And here we provide evidence that a genetically based trade-off between salinity tolerance and aggression plays a role in limiting the range of the trinating guppy on Trinidad. Now, this does not exclude the possibility of other evolutionary constraints being at play, but rather I think it provides evidence, empirical evidence from a natural system, that trade-offs are one potential solution to the evolutionary problem of range limits. Further, we went to try to find the mechanistic source of this trade-off, and we found evidence for hormonal pleiotropy. And I'm very interested in this network approach because I think it allows us to better understand how evolution might ameliorate this trade-off, and we want to continue to pursue this. And another way we could learn more about the trade-off is investigating the swamp guppy more, because they seem to be displaying this trade-off differently. And by understanding their approach, their physiological approach to it, what could that tell us about the evolvability of trade-offs and biological networks in an ecological context? So with that, thank you all for being here in the afternoon of the last day. That was a lot of results in a short amount of time, so I'm very excited to take your questions, but I also have some literature with some of the nitty-gritty details. Thank you so much. Yeah.
So, do you think there's a role for like energy expenditure energetics in Trinidad and Guppy brackish water?
A- Absolutely, that is a non-mutually exclusive hypothesis in my opinion, and I think it is definitely playing a role. So this was, as you might imagine, a bit simplified, but one thing that's really cool about these two species is that in food, there is a species food availability interaction in how they deal with salinity. So in high food, alone the Trinidad guppy grows at the same rate in brackish or freshwater. But as you decrease the food level, the difference between the salinities increases, and it does so at a rate that's more severe in the Trinidad guppy than the swamp guppy. And of course, a lot of these processes and these hormones control metabolic pathways as well. So that's, to me, is a big next step. Yeah.
I don't know much about fish, so I apologize, but are there related or maybe more distantly related species wherein you see sort of a disentanglement of this aggression to ion excretion pathway? They use completely different hormones?
I really would love to look in a phylogenetic context at this trade-off. I do have one kind of summary slide that kind of gets after this. So we reviewed, like, it was more or less a literature review to come up with this hypothesized trade-off at a physiological level. And you can see that we have literature sort of supporting these mechanism pathways across a lot of teleos fishes. So there could be exceptions, and I think that would be really interesting, but it could be a fairly conserved hormonal pleiotropy. But I want to learn more. All right, I think we should probably switch, but I'd love to talk after.